Hi everyone, and welcome to another episode from ZEISS Microminute from ZEN Microscopy Customer Center in Dublin, California. I am David Giraldo. I am applications engineer for electron microscopy. And yes, the episode for today is correlating light and electron microscopy in cryo. And please scan this QR that you are seeing right now on your screen to get the notifications for the next microminutes. Every week we are streaming these kind of episodes with very interesting topics about microscopy, different topics, different techniques. Next, for example, is on December 19, and is about characterize deeply varied defects on XRM to laser 𝗶𝗳𝗀𝗁 microscopy. Another correlations episode. Please scan this QR and sign up for notifications. Okay, let's move on. This is me. This is behind me. You can see the ZEISS Innovation Center in Dublin, California. We are located on this first floor, this area. This is the Microscopy Customer Center. This is David. And now, let me move on now to this one. This is the title for today, Correlating Light and Electron Microscopy in Cryo, Correlative Confocal Microscopy, and cryo-fixation SEM for life science insights. Let's start. Now, please, if you have any questions, input your questions in the chat, and I will answer your question after the session. Okay, let's move on. Now, starting with this, we are discussing today about cryo-microscopy. What's cryo means? Cryo came from a Greek word that states cryo or cryo with K instead of, instead of C in the Greek, in the Greek word, this is with K, and in Greek means low temperature. Of course, this is a technique for immobilizing and imaging biological specimen. Also, you can, of course, imagine another kind of specimen, but today we are focused on the cells or tissue. The important and the key factor here is we need amorphous size. We don't need here any crystallized. We will see and we'll be discussing why this is important. These techniques, the most important feature for this technique is for cryo is considered that being preserved near to native state. That's the other key factor here. We are keeping our sample in the most near native state. And it's a gold standard for life science. The purpose for cryo is localization and structural analysis. If we want to see how is the ultrastructure, what is this ultrastructure like, the localization of this ultrastructure, this is the gold standard technique for that. The other important purpose for doing cryo is keep that event that is important in development biological in the dynamic effects in glass-like state. I mean, we are keeping that state in the way that you can evaluate these effects on different states, for example, and you can have different samples and evaluate these dynamics in your samples. Brief overview, brief background about these techniques. In 2017, Nobel Prize was granted to these three persons, Jacques Dubuquet, Joachim Fram, and Richard Henderson, for the development in these techniques. Here you can see a really nice, a real nice overview of what is this kind of results. And this title states how cryo-electron microscopy provides first view of cells nucleus in natural under-Steinberg environment. That is so important, and that's something that we will discuss today. Here, for example, you can see chromatin, nuclear laminin, microtubules, nuclear pore complex, and how the reconstructions allows to evaluate this ultrastructure in 3 dimensions, and how is the organization of this ultrastructure. This cartoon is really nice. It's probably, it's the same here. You can get the information in that specific moment and what is happening with the cells or your sample here at that specific moment. For example, this guy here was in a very moment, but probably in a windy moment where, it was freezing at that moment. And the important thing, you don't want to disturb that specific moment. And that's the way. This is the technique that allows to get that kind of information. Introduction, key question asked for life science, and probably if you are asking this, if you have some experience and you want to study this, this is probably the technique that you need to answer this kind of question. And the typical question that the life science scientists did is, where do macromolecular protein machines localize within the cells? As I mentioned before, if you want to localize 3D context in your ultrastructure, this is the technique. How are macromolecular proteins matching structure organized, or how do the macromolecular proteins matching interact each other, and how the structural and position of these nanoscopic entities respond to new stimuli? That's the key question that you resolve this with this kind of techniques. This is a very good example of nuclear pore complex. And what is the result, the final result for that, the 3D structure that we are getting? This is kind of, imagine that we are collecting with this kind of techniques 2D images, but we are collecting serial section imaging that allows to create this kind of information, or electron tomography, when we are creating a very thin section of the sample, and with tilting imaging we can collect and create electron tomography, electron tomograms. Specifically, here you can see the NPC, the nuclear pore complex, and the different ultrastructure that we can acquire from that. What is the key benefits from these techniques? Almost artifact-free fixation. Of course, every technique in biological, in life science, that needs for fixations, or at least chemical fixation, that introduce any artifact, and that's why cryo-electron is superior in that context, because we preserve the ultrastructure with almost free fixation effects. And of course, we bypass challenges associated with the sample preparation for correlative microscope here. That is so important, and what is the benefits from chemical fixation? Here we can see a very good example of electron image from chemical fixation image here. Introduce certain artifacts like shrinkage, member distortion, redistribution, or an aggregation of proteins. Here you can see this image. The lipids that you can see here looks compressed here, and the difference with the cryo, you can see with that information or that ultrastructure entities in real good shape in their real native state here. That is a very good example that we want to avoid chemical fixation and what we want cryo instead of doing these kind of things. Of course, we are preventing those kind of alteration or artifact that is associated with chemical fixation. How this works and why it's so important and we can create that cryo-fixation. Okay, in regular way we have, in room temperature we have, of course, water in liquid. When we cool down that water, of course, we have crystallize. But when we have real fast conditions or the proper conditions under cool down that water, we can get amorphous water. I mean, that means we don't leave the sample enough time to get the crystal, the crystal structure that will create that crystal water. And that's why it's so important. What does mean this for biological samples? Okay, in the native state, we have water in salt and liquid. We have all this environment here in very regular way. All the water is filling the membranes and all the ultrastructure, all the organelles inside the cells. But when the crystal in water appears, that breaks the membranes, and that's something that we don't want in our samples, of course. The only way to prevent that is creating that amorphous size. That allows to get intact ultrastructure. Okay, but what that is so important, because with that sample in that condition, we can get high resolution images in 3D density maps or proteins. Originally, for example, ultrastructure analysis or isolated proteins from single particle tomography. That's the ultimate goal. The ultimate goal is try to get in a TM, in a system, in a transmission electron microscope, a very thin lamella, and tilting that lamella in different positions, as you can see here. For example, between 16 negative and plus 16, we are getting different images, tilting this very thin sample that we acquired before. And at the end, we can reconstruct that bunch of images, that set of images, and get this 3D reconstruction. We have alternatives. The answer is no, because as we mentioned before, other techniques introduce artifacts, and we don't want to study that kind of artifact. We don't know if that artifact came from our sample or the preparation. To avoid that, then this is the technique. Okay, this is an overview of the workflow. Here we have the first vitrification part. After that, we have the preparation of the thin samples, and after that, we introduce that sample in our transmission electron microscope, for example, and at the very end, we are going to get the reconstruction and segmentation and analysis for that. Then this is where ZEISS provides solution for your sample, specifically this part of the preparation. After you get the vitrification, you can use ZEISS-like microscopes and Crossbeam microscope in tandem to get your region of interest and then target that specific region of your sample. And after that, imagine and construct your bunch or your data analysis here. This is a video here. Let me change this. That shows a very fast animation from the correlating cryo workflow. Here you can see this is the sample prep. This is the confocal microscope, and this is the Crossbeam. In the prep table, we have all the kit for manipulate the sample. We have grid bus, correlated cryo holder, and other adapters that goes with this kit. Here you can see this is the grid vitrified after we got the vitrification for different kind of samples or different kind of approach for that. We are now loading our samples, our grids, in the Lincoln and goes directly to the LSM. Here we have the Lincoln cryo-correlative. A previous episode, if you want to see more detail about this one, you can go to a previous episode where Amira, my guru, our colleague, our guru here in LSM, describe how to screen your sample and how to get your best sample, because this is so important. I mean, you don't want to waste time preparation. If you don't have the right preparation and you go directly to the electron microscope, you are wasting time. With the LSM, you are preventing of that. You are screening your sample. You are getting a real good detail from your samples, and you can be ensured that you have the right sample before you go to the electron microscope. After that, after you collect that kind of information, then the sample is ready now to load in the proper sample holder. And with the cryo vacuum transfer, you go now and you collect now. This is under liquid nitrogen. Always this is under liquid nitrogen. This is the only point that the sample is not under liquid nitrogen, but it's this is isolated and ready to go to the preparation chamber here. Now this is very fast, and you can now go to the preparation chamber. Here you can perform the sputter coating. That allows to prevent any charging effects. After that, you are going to charge in the main chamber. And now here you can correlate the information that you get from the confocal microscope and now target that specific region, that specific cells that you want to create acquiring imaging. And this is how the imaging or the process looks like for TEM lamella preparation, for on-grid lamella preparation. Okay, we are creating this lamella, and now this lamella is ready for cryo-electron tomography, which is performed in TEM. Okay, this is an overview. This is a real cool animation. Highlights seamless cryogenic workflow across the multiple modalities, optimal sample protection, high resolution fluorescence, high contrast volume, and you can target your specific lamella in the TEM application. Of course. Now, this is another cool animation, how the process is. This is the FIB and the electron gun acting in tandem. You are milling and acquiring imaging. After that, you can create the lamella, and after that, you can go to the TM and acquire in different tiltings orientation to get the 3D tomogram here. Okay, this is very cool animation, but how this in the real life, in the lab? Now let's move on to the lab and see how this looks like. This is our sample holder here. Here we are seeing the inside part of the chamber of the crossbeam here after we create or we collect all the information for the LSM from our confocal microscope. Then we are load the sample inside of here, and now we are seeing how the lamella preparation and the 3D reconstruction, in the same sample in the same site can be performed. Okay, first we are collecting some image here. We load the sample. This is how the grid look like. But now in this big area, okay, it's in context of microscopy, it's a big area. Where is the specific target, the specific site that I want to mill that I previously acquired? And so with an electron, with the confocal microscope, now I want to reach that point. Okay, now we are setting the parameters for our sample here. Okay, we are saving this image here. This is our image. We are naming this image. After that, we can open our ZEN Blue. This is our project. This is all the information that we already collect in the confocal microscope. This is how this project looks like. And now we are hitting snap and acquire the image. ZEN Blue take over the Crossbeam imaging sample process, and now we are collecting imaging, and we have full control for the imaging and for the stage at this point. Okay, now we are aligned. We click on Align System. That allows to only overlap the imaging here, and we are looking for feature that allows to correlate in a proper way this image or these two jobs here. This is an electron image, and now we are overlapping with the confocal image here. Now we are selecting the alignment mode. This is translate and rotate only. We only click here and starting the alignment, rough alignment. After that, we are getting we are performing the final alignment. For that, now alignment mode is translate, rotate, scale, and share. We have two different pins that we can place once we are happy. This is our second pin. Now we can correct the sharing because, of course, we have different instrument here, different scales, and that's create and sharing here. Now we are ready and we are happy with our alignment. You can see these features fit very well with the confocal feature that we already collect here. Okay, after that, then we are ready to navigate through our samples based on our confocal information. This is our region of interest. Now we double click on that, and that's drive the stage to that specific location. And here, of course, this is our cluster of cells or the region of interest that we identified previously in our confocal. And now we are going to perform the milling part. We are now, put our FIB in on. Once this is ready, now we are going to work in Smart ZEN and Smart FIB to acquire that information. Now we need to acquire a FIB image. This is our FIB image. We are in the coincident point. This is our coincident point. Only keep in mind that everything in the FIB will look like upside down. And now we are positioning the element. This is the FIB element to start the milling process. Okay, now we are going to adjust this element. This is the very first element over the region of interest. This is the region of interest. This is the cluster of cells that we previously identified in our confocal. Now we are setting the condition for the milling process, 300 picons. And instead of cross-section, we are going to get serial section imaging. That allows not only doing the milling, but also at the same time milling and acquire imaging that allows to create 3D reconstruction at the same time that we are getting the thinning process or the first thinning process of the lamella or the very top down part of the lamella. Okay, now let's move on. And let me show how the serial section process starts. Remember, the very first process is the milling process of the surface of the sample, of our sample, and this allows to create also the serial section imaging process. Yeah, this is another example, not the same sample, but yeah, it's the same process. And now we are getting our serial section imaging. This is the element, very similar to the previous one. And at the same time that we are milling, we are collecting imaging. That is a real cool feature because you are getting information. You are not only milling and wasting time and seeing what is going on with my sample. Here we are seeing what is happening with our sample, and we are collecting even very, important details. Of course, we can correlate that information because we already do the alignment of the system, and all this information is in the context. And here you can see an example of that. This is our previous correlation, our rough correlation that we already see. Now we are acquiring the same image that you previously see that was acquiring in the milling process. And now let's see how this information looks in the context. Okay. Here you can see the cells. This is a cluster of cells that we identified previously. And now you can see that image in the context of the cells. Okay. Yeah, we can move the transparency here, and we can see how the details of, for example, mitochondria here looks like. Yeah, this information from the confocal and all the information here is the nucleus, the membrane of the nucleus here, and other organelles, other mitochondria here, and you can see how the information correlate. And that's very, very helpful. I mean, try to do this without the context. I don't know how can do it without, this technology. Yeah, that's pretty much that I have for today. The final result for that. This is how the final result of that kind of information look like. We can navigate through our sample, through our detail of that sample. And last but not least is tomorrow we'll have a workshop. If you are interested in more details about this one, please scan this QR that you are seeing your screen. From Jupiter, Florida, we are streaming this event, this ZEISS correlated cryo-fixation SCM workflow for cryo-Lamella preparation and volume. We have some very nice talks. Joe Mallory and Chris Bryan will be showing very important details, very good information. Amira and I will be doing the demo part here from Dublin, California. Please scan this QR and you can have more details about this nice event for tomorrow. Okay, that's pretty much that I have for today. Thank you so much. And let me now move up again to this. And let me see if we have some question in the chat. Okay. Okay, so far we don't have any questions. Okay, if you don't have any question, please don't hesitate to contact me or contact anyone in the teams from Science Microscopy Customer Center in Dublin. We are happy to assist you, provide any help. Thank you so much for being here, and we'll see you in the next one. Bye-bye.